Hi, my name is Caroline Pitts, and today we'll be talking about positive inducible regulation of prokaryotes. Here is what the entire process looks like, and we'll be breaking this down step by step. First, the meaning of positive inducible regulation can be broken down with these two definitions: inducible operons and positive control. What this means, put simply, is that the default state of gene transcription is off. However, we want this site to be turned on, but the regulatory protein cannot do this by itself. After an effector is present, it will bind to the operator, turn on the gene, and transcription will occur. Please be sure to review these other important definitions, as they are crucial to understanding this process. Here we can see a diagram of our DNA, including our promoter region, operator region, and 3 structural genes. Before these genes are coded, RNA polymerase binds to the promoter region and will travel the length of the operon, but this process can be halted by the operator depending if it is bound with the regulatory protein. In this case, the regulatory protein is called an activator protein and must have a small molecule bound with it, and are both only active when bound to the operator. The addition of a small molecule is known as inducible control. Now that the small molecule is attached to the activator protein and the activator protein is bound to the operator, RNA polymerase is finally able to move across the operon segment and the structural genes are transcribed. Now let's take a look at a real-world analogy at how this process would work. This is my boyfriend Danny. As we can see, Danny is only truly motivated by food. Today, I need Danny to clean up the apartment, so I'll need to tell him to do so and provide a burrito to make sure the tasks are complete. Transcription would be Danny cleaning the apartment, the activator protein is me telling him to do so, and the small molecule is the breakfast burrito. First, we will see what happens if I tell Danny to clean up the apartment without motivating him with a breakfast burrito. Hey Danny.
Huh.
The apartment looks pretty dirty. Can you clean it for me?
No thanks.
Here we can see that without a cheesy breakfast burrito, Danny will not let me boss him around and clean the apartment. Similarly, without a small or effector molecule, transcription will not occur, which would be cleaning the apartment, because the activator protein is not stimulated. Now let's see what happens if I tell Danny to clean the apartment and I bribe him with food. Hey Danny, the apartment looks pretty dirty. Will you clean it for me? Here we can see that the small molecule was able to stimulate the activator protein, which allowed transcription to be completed. In other words, Danny cleaned the apartment because I gave him a burrito.